All right. So the next part of the lecture, I'm going to talk a little bit more on the published examples, and it's not a very detailed introduction. So basically, gives you some example that what this RNA sequencing can help us. Right? You may just think it only sequence the RNA. I know it, but there's a lot of very creative applications that you may feel very interesting. Okay, the reason I talk about this example is this is, I think, the first RNA sequencing experiment being published, and from Bavaria's group, and in May of 2008. There may be some other individual studies earlier, but this is the one that has the largest impact for sure. Okay, and the title of that paper is Mapping and Quantifying Mammalian Genome. What they did is, this is one of the first, and what they did is. The experimental detail is they use a tissue, which is mouse, liver, muscle, and total brain tissues. And they did 2 technical replicas and 2 biological replicas for each individual tissue. And the sequences were polyadenylated, and they used the Illumina technology of 25 base pair arrays. Okay? So this is a very early study. At that stage, this is the state of art. So a lot of things that I'm talking about in this paper, you may think it's so straightforward, or sometimes it's even laughable, but that was 3 years ago, and the technology was a 25 base pair. Now we know we are pushing 125 base pair, and for some of the platforms, and for 454, it's even 400 base pair. So technology development really improved a lot. And what they did is, even though the reads are pretty short, they did 41 to 52 million mappable rates for each tissue. And that is a lot of information back in those days, especially when you work on 25 base pair arrays. We know that with this length, a lot of reads are not mappable back to the reference genome. Okay, remember we have a mappability. I think there's a kind of a percentage of mappable regions that can be identified. And so they did really, really deep sequencing. And I also suspect they didn't pay for the experiment. The Illumina paid for it. And the reason that I say this is one of the first RNA sequencing experiment published, because I know they must be on certain level of pressure. Because this paper was accepted in May 30th of 2008, and another paper comes out from Science early June. Then they sequence the yeast genome. So you can see that they have some battling things, but I like this paper a lot. And some kind of results on that is they have 90% of unique mappable rays fell within the known exons. So this is one of the statistics in earlier days that people are interested in. So when I do the RNA sequencing experiment, when I identify so many rays, what's the percentage of the rays are in the gene? Or if they are in the gene, what's the percent are in the exon regions? Because hypothetically, those are the regions we're interested in. And their percentage is 93% exon. This is one example. And overall, it's over 90% in the exon regions. And one of the reasons that they can achieve this high percentage, I think, is they did poly A selection. So they focus the most on the mature messenger RNAs. But if you do other type of assay, if you use random primer and ribosome depletion, there's no way you can get such high percentage. But this is what they get. Okay? And they said they were able to detect and quantify differential expression. And so you can see it sounds like pretty straightforward. This is 3 different tissues: brain, liver, and muscle. And this is a muscle-specific gene. And they went there, they see there's very strong signals in that region where there's nothing in the brain and the liver. And they also, just based on this, they concluded that it's high sensitivity, very little identified in brain and the liver. So because this is muscle-specific gene. Okay? And another bigger contribution of this paper is this is the first to introduce the so-called RPKM, which is the reads per kilobase exon model per million mappable rays. RPKM measurements to quantify gene expression levels, which we will talk about in the next couple of slides very extensively. And they also see very high reproducibility between samples. So you can see that this is the technical replicas, this is the brain. This is the first batch and second batch, and their signals can achieve to R-square value 0.96. It's really, really high. And this is another very interesting figure that I think affecting a lot of later study designs. And they found that the dynamic range is high. And we kind of know it based on the review papers. But what they did here is they used the spike-in information from Arabidopsis DNA. So they are made sure that those DNAs are not in the mouse genome. So it shouldn't be confused with the alignment issues. And then they did this back in and put in different doses. Okay. So what they found out is this is based on 100 nanogram of messenger RNA, and this is the number of reference transcripts they put into the cells. Okay. So you can see this from 10 power 4 to 10 power 9. I'm going to use this 10 power 9, and this is the RPKM value they have identified. And you can see that it's very, very nice linear phase. So you can see the more RNA transcripts they put in, because this is spike in, so they know the answers they put in, and the more signals they will get. It's very, very nice linear phase. Another information that I think we need to take advantage of this data is it tells you a good estimation of how many reference transcripts are in the samples that you are identifying. Okay, so if you identify something, you say, I have observed one RPKM, this gene expressed in the level of 1 RPKM. Okay, that is equivalent to how many transcripts in the samples you have, and based on this, you can really look at it's equivalent to 10 power 5 of transcripts in 100 nanogram of messenger RNA samples, and which is very important information. Okay, and this one is even more difficult to understand, but it's very useful. And they found robustness of RPKM measurements as a function of RPKM expression level and depth of sequencing, which means that the deeper the sequence is, the more accurate your measurement will be. Okay, that for sure, of course. But what they got the figure here is you can see there's multiple lines here. Okay, I'm going to explain that. If you cannot see from back, let me do this. Let me blow this big so that we can all see this, because I think this is an important figure to look at. They should be able to see this. Okay. So what this tells you is X here is the total number of mappable arrays from less than 1 million mappable arrays to 41 million mappable arrays. So you can see this is an increase. And for this one, Y here, it tells you the fraction of genes within the plus minus 5 of the final value. Final value meaning they did the spike in information, so they know how much material they put in, and they want to know whether they got the real measurements. Okay, so then let's look at this black line here. You probably just see a angle here, and this is for the genes that is larger than 3,000 RPKM, and actually there's not too many 3,000 base RPKM genes. It's very, very highly expressed genes, but you know that once, even though for the genes of the expression level is this high, even we didn't sequence very deep, you can get very accurate. Okay, so the fraction of genes within the plus and 5% of final value is 100%. However, once you get to this one, which is the blue one, this is 300 to 3,000 RPKM, and you can see that this is start to tweak a little bit, and you can get 100% accuracy once you got about 8 million mappable arrays. Okay? And next is 30 to 300 RPKM. And even though you got 41 million mappable arrays, you probably got pretty close to the final value now. And if you are looking at about 15 million mappable arrays, and you have about 80% of the genes, their expression level you can detect pretty accurately. And if we talk about 3 to 30 RPKM, and this is the performance. So the point here is the deeper the sequencing is, the more accurate your result will be. And but keep in mind, this is 3 RPKM is already a pretty big number. So you probably, based on this type of measurement, if you think deeper, playing with the real data, you can sort of estimate how accurate your final result is. Okay, so I really want you to go back to read that paper, and there may be some questions from that in the exam, just some hint. So just go ahead to study that. I think that will be very, very useful. Okay. The second study I want to introduce a little bit is from Chris Birch's lab, and also published in 2008, and to study alternative splicing. So what they did is they did 10 diverse human tissues and 5 epithelial or breast cancer cell lines, so 15 individual tissues. And what they did is now this is 32 base pair arrays, a little bit longer, and 12 to 30 million arrays per sample. They also did poly-A selection, and they found that 60% of arrays mapped to genome. And actually, for the variable study, I didn't see this statistics. So they didn't tell us what's the percentage of arrays were mapped back to the genome. Maybe they did. I didn't catch it. And they also found the 4% of the arrays mapped to the junctions. And this is a 32 base pair arrays. It's very, very short. It's pretty good that 4% of the arrays mapped to the junctions. And they also found that read density is 100-fold higher in exons than in introns or intergenic regions. So you can see in the exons, by average, it's 12.3 RPKM, but for introns and the intergenic regions, it's much, much lower, okay? Which tells us that their experiment did pretty well. Coming to the science part of it, and this is the first time that we are able to really profile alternative splicing in the genome scale. And they found that 92% of multiple exon genes were estimated to have multiple isoforms with relative frequency of minor isoform is more than 15% in one or more samples. So what this tells us is we know that 94% of human genes are have multiple exons. Okay, among these 94%, 92% of them has been alternative spliced one way or the other across 10 different tissues. So this really tells us alternative splicing is a much more universal event and comparing to what we have thought before. We think probably 50%, more than half of the genes are alternative spliced, but the result is no. 92% of multiple exon genes, they are alternative spliced, and this is really remarkable. That really tells us that if you are looking of a science direction, and probably alternative splicing is a good direction to go, because we don't know too much about it, and there's so much lower fruit to be picked over there as well. And splicing patterns are tissue-specific. This is another conclusion. They found over 22,000 tissue-specific alternative splicing events was identified. So over 60% of alternative splicing events are tissue-specific. And what they did is they did a cluster against the different tissues, okay? And you can see these are for the individuals, the same tissue of individuals, and these are all on the brains, different brain lines. Okay, and these are on different tissues, so breast, fat, and lymph nodes, and the colon and testis, and liver, and 10 different tissues. So you can see clearly that the variations that within the tissue, but across the different individuals, but they still cluster together. Well, for different tissues, they kind of varied greatly, but for this is another brain and the cell lines, and they cluster together. And they found that variations of alternative splicing between individuals is 10 to 30% of events, and between tissues are 47 to 74% of the events. Okay? So this is another paper that using RNA sequencing experiment that first time we are able to understand how diverse our transcriptome is, and in terms of alternative splicing. Over 92% of multiple exon regions are alternative splicing. Multi-exon genes are alternative splicing. This is remarkable. And another study is to study gene fusion. So what is gene fusion? It's a fusion gene. It's a hybrid gene formed from two previous separate genes. So you can see this is one chromosome, this is another chromosome, there's one gene here, the other gene here, right? And ideally, that in the real individual, I mean, in the normal individual, and you will see this gene transcribed and this gene transcribed separately. However, because of something happened, and on the DNA level, these two chromosome part, they kind of connected to each other, okay? So the end result here is you will be able to see some of the transcripts and started from this part and then end towards this part. This has been observed a lot in cancer cells. And the point of this figure is that if you use a pair-end arrays, and that is really helpful to identify this type of events. Okay, and this paper was published in Nature in 2009 to study gene fusion. So this is one example. So you can see this is the EGR gene, this is another gene, and they should be separated, but somehow they connected together. And you can see that here it uses the so-called single-end technology. They only use one read, not the pair-end two reads. But you can see that at this joint location, and there's you can see very clear evidence that the gene fusion occurred. Of course, it's easier to say and easier to look at from this point of view, but before you do the alignment and everything like that, it's pretty difficult to find this type of patterns. Okay? And this is the experimental procedure they did. Actually, this is a quite unique study. It's not a straight RNA sequencing. What they did is a two-step process. The first step is they used long reads, 4x4 reads actually, and they just did RNA sequencing using 4x4 reads, and they identified some regions because they are so long, and it's much easier to do alignment. And if one end is in one exon, the chromosome, the other end is in another chromosome, right? They identified a few potential fusion events, and based on the longer reads, and then they designed the targeting procedure for the later study, only focus on this region, but on shorter rates, but much more deeper rates. So give you a lot of greater evidence. So this is the first paper published study gene fusion using RNA sequencing experiment. So the experiment is not really can be reproduced by a lot of groups, right? And not in terms of science-wise, but in terms of the experimental setting-wise, it's a pretty difficult thing to do. But now, because we have the pair-end protocol, and there's some single ends, the reads become longer and longer, going towards 125 base pair. So to study gene fusion, and it certainly become possible. And next time I'm going to talk about that, and there's a couple of computational tools already being developed to study gene fusion from both single and pair-end data. And you can use both single and pair-end approach. Okay. Next study I want to introduce a little bit is the single-cell approach, and this is published in Nature Methods in 2009. And it's mRNA sequencing whole transcriptome analysis at a single cell. And what they did is that they did get one human blood smear and oocytes, okay, one cell. And they used solid protocols. They identified over 75%, over 5,000 more genes than using microarrays. So this tells you that with the proper experimental design and a single cell level RNA sequencing, can be much more sensitive. They identified over 1,700 previous unknown splicing junctions just in one single cell. Okay? And they also found 8 to 19% of genes express at least 2 isoforms in the same cell. That is kind of amazing. So when I show you the previous figure that you see the observation of both 2 isoforms in one plate of a cell, but that is a lot of cells. So it may be possible that the isoforms, some isoforms is in this group of cell, other isoforms in that group of cell. But here, what they conclude, they found that almost 20% of the isoforms, and they found both 2 isoforms in the one cell. So that really tells us very good regulation is going on there. And the next point here is they found a good reproducibility between 2 cells. So this is very amazing. So this is the first cell. This is the second cell. And you can see that within these 2 cells, this achieved so good reproducibility. It's really, really amazing. And the R value is 0.986. Okay? Well, you probably don't know how difficult this is. And actually, we had some experiments working with the UCSC group. They also work on the single cells. And our correlation between 2 cells, 2 individual cells, are 0.7, 0.6. Okay? And I don't know how they achieve this high sensitivity. Yeah?
What kind of cells were they?
I have no idea.
These are oocytes.
These are big.
You expect them to be fairly similar, especially if they're from the same animal.
Yeah.
You may have taken 2 cells from like that, and you don't expect those 2 cells.
That's a good point. I don't. Yeah.
Brain cells, you may have gotten an astrocyte and a neuron.
Mm-hmm. Yeah. I suspect what they did is 293 cells, so the cell lines, like individual ones. And but this is really, really amazing, and I was very amazed by this result last night where I was preparing for the slides. And the next application for the RNA sequencing is genome annotation with the RNA sequencing data. And the RNA-seq data sets improve the genome annotation in plants, animals, bacteria, especially for non-model organisms. And you probably don't see this list from the back. Let me read it to you. I cannot really read it because some of the words are not really English. The first one is watermelon. And I see the oilseed rape. And I don't know any of this. And there is the wine grapes somewhere here. So you can see that people started taking advantage of this technology and sequencing a lot of things on this. But the point is here is not just for the sake of sequencing, right? They want to annotate these genomes. So we never had technology before. I mean, to sequence most of these species, probably the DNA sequencing has already been conducted, and so we already know their reference genomes. And but if it's not, we can do it, right? We know that BGI has a lot of machine sitting there and want to sequence something, right? And so last week we were in a conference, and there's one speaker from BGI, and the slides. BGI is the Beijing Genome Institute. They have a couple of hundred HiSeq machines. The first day, Illumina released the HiSeq, and they bought probably over half year of their production capacity. So nobody else can get the machine, and they bought 120 something of that, and so make Illumina busy for half a year, cannot get anything else shipped out. But they have really huge sequencing capacity, and the slides they show is one slide is what we are sequencing, the dinner, all right? They have different the salmon and the chicken and all this different pretty delicious stuff, and they sequence it. The zoo, the panda, the gorilla, and it seems that every living organism they are sequencing it, and I don't know whether it's true or not. But anyways, with the RNA sequencing part, so the DNA sequencing, you can get their reference genome in a reasonable okay way. But For the RNA sequencing, it helps you to do the genome annotation, right? You know not only the sequences, but also which part of the genome has been transcribed, and that is really important scientific questions. Okay, this is especially important for non-molecular model organisms, and it may need to couple with DNA sequencing, or sometimes require de novo transcriptome assembly. And there are some tools that can do that, including Trinity. And I hope I will talk about that a little bit. Okay, try to read that paper. Never understood, and assembly in general is beyond my head. But I will try my best to summarize that. And there's another tool published that you can take a look at. The limitation for this type of approach to the annotation is the genes are expressed at a low level, will be represented by few reads, and may not be, maybe only partially covered. So, which if you have a good reference genome already, that's fine, and you just see low signals there. But if you don't have a good reference genome, you need to do the assembly first, the transcriptome assembly first. And for these genes, you really need to sequence extremely deep to be able to do that. And most of the published studies, they were not very deep yet. Okay, but I suspect that with the increase of the capacity of the DNA sequencing technology, and this can be measured more and more accurately. And some people also, this is fall into the metagenomics type of research area. And there's a March 2008, there was a paper published in New England Journal of Medicine, and they already used this in the clinical application. So the story here is they found the 3 patients who received organ transplantation from a single donor on the same day died of this febrile illness 4 to 6 weeks after transplantation, all right? And they want to figure out what's the reason for that. And it's really not towards the scientific discovery, right? They want to know the reason. And I think that this is published in early 2008. So probably the experiment was done in somewhere in 2006 or 2007, at the time that Illumina is not there yet. For those of you who doesn't know, the first commercial sequencer is Roche 454. It's released in 2006. Illumina 2007, Solid 2008. That's why that you see the huge imbalance of different market share from different platforms, because some platforms get into market 1 year early. They got a lot more Nature Science papers compared to other platforms. So people just follow that. And that's one of the reasons that Solid has a late start. But for this particular application, I suspect they use the 454 applications, and the reason for that is the number of sequences they are using is remarkably low. It's only 104,000 sequences, but among these, they identified 14 represent an old world arenavirus. Okay, and they confirmed the presence of this virus in kidneys, liver, blood, and spinal cord fluid of these recipients. This is using the RNA, the PCR assays, and then their conclusion is the donor has some virus that carried into the recipient and caused them die. And the conclusions here is powerful to discover pathogen. And I heard about one study, but I didn't find it in the publication. Is some people identified the virus in cancer cells, and talk about that is one of the reasons some of the cancers can be metastasis. And but I haven't found that publication. If you can find it, feel free to send it to me. Okay. Another major application is small RNA profiling. And this is the first, I think it's the first paper published about the small RNA profiling. And what they did is they used the human embryonic stem cells before and after differentiation into these embryoid bodies. Okay, so what they concluded is 52% of the sequences, the RNA molecules they identified, are actually microRNAs. And of course, there are some other RNA species. And they identified 334 known and 104 novel microRNAs, and some of them are differentially expressed. So nothing too unique about this study, but just to tell you that using RNA sequencing technology, we can identify novel small RNAs, which is important for a lot of applications. Before we get into analysis, there's another thing that I want to talk about in terms of application that I haven't prepared slides on, is the RNA editing is another thing that potentially is important, and only with RNA sequencing technology that you should be able to identify that. And some other applications that we have seen that one paper published in New England Journal of Medicine that they identified novel genetic variations in the RNA sequencing samples of the cancer cells, and that is kind of a pretty big discovery back in those days. So there is a lot of very creative RNA sequencing applications, and I think you really need to use this technology creatively to be able to help your research, not just only the routine analysis. Okay? So now I'm going to switch gear a little bit and really talk about data analysis part. And for the RNA sequencing, the biology is interesting and straightforward, but for the analysis part, it's not. There's a couple of challenges in the RNA sequencing data analysis. The first one is the sequence alignment. We have spent almost an hour and a half to talk about alignment in lecture 2, but none of them are RNA sequencing alignment. So what's unique about RNA sequencing? So we know this is let's say this is a DNA, this is the exon regions, there's a lot of intronic regions, okay? And at a single breakpoint here, and there's one read that covers this region, and using the traditional alignment algorithm, we cannot map these reads back to the genome, because none of our aligners can take care of large, big gaps, which for the introns, sometimes it goes to 10,000 base pairs. So there's no way we can find it. There's need to have special strategies to fix this issue. And the second challenge is transcriptome reconstruction. And you can see that this is, let's say this is one gene, and at this gene that you observe these kind of signals using RNA sequencing experiment. But if this is a reverse problem, if you see these RNA sequencing signals and you want to infer what kind of gene structures the regional samples are, it becomes very, very difficult. And the third type of challenge is expression quantification, right? And we will get into that a lot deeper today, which you can see that for this particular gene that there's so many reads that are there, and in this region there's the same amount of reads, but how do you quantify this information, and how do you compare different genes? And that becomes the issue. Sometimes it's not even possible. Okay, let's go to the sequence alignment part. Okay, and this is a slide from lecture 2, right? So just a very brief summary of the alignment algorithm in general, what kind of category they fall into. The first one is a hash table-based strategy, and we make those dictionaries and in the human genome, and then quickly go through that and do Smith-Wortman and or dynamic programming to find the remaining part of the reads, if you still remember that. And the problem of that is can be slow, and the index sizes are pretty big, right? And there's another type of strategy. It's a suffix tree or prefix tree type of strategy. So you can see this is the prefix tree, this is suffix array, and these are not really widely used because they hold a lot of memory. So it's just too big. And most people are just using this Morris-Wheeler transformation. And so these are, in terms of alignment on the DNA end. And for the RNA sequencing alignment, there's 3 major challenges. The first one is short reads. Okay? We have only 36 to 225 nucleotide per read. And you cannot really use BLAT or things like that to do this type of alignment. And the error rates are considerable, and the major difficulty is many reads span axon-axon junction. So we have this point. This is a junction, so there's a reads here, and it's really, really hard to find the proper alignment for that, because aligners are so alignment should be conducted both on genome and the transcriptome, but short reads aligners are no gaps allowed very small gaps, and neither of these will work on the intronic regions because introns are just so big. And so the strategies people are using for some earlier studies, that people just forget about those junction reads. I'm not going to take care of it anyways. Let's just using map to the genome approach. And this approach is very simple, and sometimes it can be effective in the earlier studies if their goal is just trying to do the annotation and the quantification of the gene, right? They are not very much interested in the gene structures and how splicing occurs. And for the earlier studies, to identify those reads across junctions is not possible anyways, because those reads are too short. Right? It's only 25 base pair. There's no way you can have reliable alignment across junctions. But the problem here is whenever RNA read spans the axon boundary, part of the read will not map continuously, and that read is completely failed. And the real RNA sequencing, and for the RNA sequencing experiment, and then people take several different strategies. The first strategy is they use something called the unspliced read aligner and to read across the transcriptome. Okay, so I'm doing an RNA sequencing experiment, and I can align my reads back to the genome, but since if I'm working on the human samples, so we have so many annotations of human genome, let's just make the transcriptome as my reference, and I will just map my reads directly to the transcriptome, and that will work, right? And our algorithm of the same as the previous presented, and bow tie, BWI, shrimp. It's just the algorithm is the same, just where you map to is different. Instead of mapping to the genome, you are mapping to the transcriptome. So that's the point. And again, it doesn't allow large gaps. Ideal for mapping reads against the reference cDNA database for quantification purpose. And it's not good for identifying novel transcripts. We know that because we are aligning my reads against already known transcripts. And the RNA sequencing strategy, alignment strategy for the unspliced ones, just to compare this a little bit. For the BWA-based approach, it's very efficient when exact reference transcriptome is available. Okay, and this is the property of the BWA. Remember, when we go through this BWA calculation, we never talk about how they go through the mismatches and things like that, right? And actually, it becomes quite complicated because I didn't understand that. I didn't go through that. And but the point is, once you have more variations in the your query against the against the reference, and the BWA or bow tie, their alignment efficiency decrease dramatically. So most of these algorithms will only allow 2 mismatches. If you have more than that, and they really have a hard time to figure out. And performance decrease exponentially as the number of mismatches increase. But for the seed extension approaches, when only reference transcriptome is when only the reference transcriptome of distal species is available, and seed extension approach will dramatically increase the sensitivity. That happens. It may not happen in human or model organism, but some other things that you study a species and the reference genome is not available. Sometimes you want to map it against a relative close species. And at that time, don't use BWA at all, or don't use any Boris-Wheeler transformation because the sensitivity is too low. Then you have to use the seed extension approach. I think BFAS or some other approach is a good choice. Okay, and the study suggested that map mouse reads against a rat reference transcriptome, 40% or more reads aligned at a cost of 7 times more compute time using seed extension approach comparing to the Borel-Wheeler transformation type of approach. And the increase in sensitivity when aligning reads to a polymorphic region of the same species. So if some region, they are kind of very different and a lot of SNPs there, and probably you don't want to use a BWT-based approach. Okay. One example for this is we know there's so many different rat strains or mouse strains. Okay. If you study something that you use Bernoulli, and you probably are in the good shape because the reference genome is Bernoulli. But if you use other rat strain, you may want to evaluate that a little bit more. So in our practice, we also observe the same thing, that BWA will produce a much lower number of rays comparing to BFAST. So those are the choices that you have to consider. Okay. The second type of strategy is the real strategy, right? The first type is just use your whatever aligner you are using, but map it against the transcriptome. But for the second part, and there's a spliced aligners. There's two different forms. The first one is called the exon first method. So in brief, as you can see, this is one exon, two exon in the RNA that connected together. And when you detect so many different reads, some black goes to exon 1, and white goes to exon 2, and there's some kind of mixtures, and these are junction rays. So the general approach for this is you first just map your rays against the genome. Don't worry about the RNA, okay? So you find some of the rays mapped here, others mapped here, and which is perfect, and none of those hybrid rays will be mapped because they are across junctions. And next stage is you further work on the remaining rays. So the way to do that is people just separate those unmapped rays into smaller chunks, and then each individual one individually map it and then further connect it. So that some people is doing that. The second type of strategy is called the seed extension method. So what they do is they don't really do the first thing. They don't map things against the genome and they're trying to identify the exons. Rather, they initially they will break every single read into smaller pieces and then map it and then connect it. So this is the two general approaches. Okay. So for the exon first method, which is this method, go a little bit deeper. And there's several available tools, and the most popular one it will be the top hat, which I will talk about in the next slides. But you can see that when people start to work on this area, they struggle on their names, right? There's two tools. One's called map splice, the other one called splice map. It's kind of interesting how much difference they make, right? And the two-step, this is a two-step procedure. The first one is map reads continuously using unspliced read aligners against the genome. And the second one is for the unmapped reads are splitted into short segments and then aligned independently and join them together. Okay? And the good thing of this approach is the efficiency the efficiency when not too many, it's efficient when not too many reads are into the junctions. Okay, and the second step is computationally intensive because you have to break all the remaining reads into smaller pieces and then separate them doing alignment. And you can miss reads across the exon intron junctions because of this type of alignment. So now let's talk about top hat. Okay, this is a software package that is, for some reason, this is identified splice size and by larger scale mapping of RNA sequencing rates. The reason I talk about that is the first one is it is very popular. It's the most popular approach for the RNA sequencing alignments. The second reason for that is it has some upstream and downstream tools that connected to that top hat very well. So some other algorithms for the downstream analysis, for example, isoform identification, they directly take they were based on assumption of your alignment was done by top hat, and then the application become much wider. Okay, so a splice junction map algorithm must be able to identify reads that may have only a few bases on one side in the junction, and or else that the junction will be missed. So for example, you got this one axon, two axon, and this one read cover here. Okay, if your read is long enough, like say 120 base pair also, you probably is in good shape. You just break the reads from the middle and then align them separately, and you can join them together, right? But if the read is like this, you probably will run into trouble, because this end, you're in good shape. You can align it. But this end, since only a few less than 10 base pairs are touching the other axon, there's no way that you have enough specificity to map it to the closed axons. So it becomes really, really hard because this part is short. And we don't really have control that what RNA sequencing, what sequencer did, how they really identify this. So this is doable, but this is really, really hard. So, and for this type of scenario, only increased read length won't help either. Okay, so this is the top hat. Quickly go through it, and actually it's very straightforward. What you'll do is you'll map your reads to whole genome with bow tie, okay, and just map your reads to the whole genome, and then this is your results. Or you see the clusters, and the next step is you assemble these align the reads here, assemble the consensus of covered regions. So basically, you want to see this region are enriched of my reads, and those are potentially the axon regions, right? Because we already observed so many different small fragments there. And next is you want to generate a possible splice site between neighboring axons. And this is one example. So you see, this is one border, this is another border. But sometimes, just based on the first level approach alignment, you won't directly touch on the axon splicing sites. It's very rare to do that. So you have to do some extension of the region and to find the gene sequence that representing the splicing. So we know for the biologists, people know that the splicing site is GT from this end and AG from this end. Not 100%, but most cases it is. So taking the advantage of that and searching for these regions and trying to define the axon boundaries. And the next step is, based on this, you construct your new axon models, and then what you are going to do here is, for the remaining reads that was not mapped in the first stage, and then you'll map those across these junctions. So that is the way that people are doing it. And for the unmapped reads, so there's one information I want to point out here is they are not indexing the reference. Rather, in this step, they are indexing the unmapped reads and trying to map it against the genome. But those are trivial issues. And the general idea of this top hat is you got the RNA sequencing data, you align them against the genome, and then you figure out which part of genome transcript become the axons, and then identify the boundaries and further align the remaining reads to these regions. So this is what top hat is doing. The potential problems of that is axon consensus. So for this step, once you map all the reads across the genome, you get all these regions, and you think this is the axon consensus. But how accurate they are? So this is one of our data. You can see that this is one gene from different axons, right? And this is a bunch of signals. I really don't know what that is, okay? I'm not sure those are real axons, because these kind of things that we see all the time across the genome in the intron regions. It could be the misalignment, it could be our analysis problem, and it also could be that some of the signals were before the splicing occurred. So this is truly the intronic region, and eventually will be spliced out. So everything is possible. So if I use this information to construct my axon consensus, I'm not sure that will work, and that's why I don't feel very comfortable with that. And the misalignment regions can also affect the performance. Okay, and very quickly for the seed extension approach, and to be honest, this is the first time I heard about that, and you can just go to look for the papers. But the point here is, before, in the first step, what they do is they break the reeds into short seeds, into small pieces, and then they align them independently and trying to join them together. So that's the general strategy. And people are talking about this will increase the sensitivity, but I don't have the data to support that. And sometimes this one arm may not provide enough specificity for alignment if this arm is too short, just like the problem I described earlier. Quickly, that this is our strategy to do that. I'm not saying this is the good strategy or the perfect strategy. This is no perfect strategy. It's just based on the considerations we have, this is currently what we are picking. So all these different steps are, you can see that this quality control information can do it, but in the alignment part, so what we did is we got a sequence, and then we first align to the genome, okay? And then we build a junction library. So for every single possible junctions, so you got an axon here and the axon here. They don't have to have the evidence connecting together and the axon there. So what we do is we build a junction. This is the potential junction, and this is the potential junction. And this is the potential junction. So every single axon that has the potential to connecting with the downstream axon to certain lengths, okay? And that would be a pretty big junction library, but we only need to do that once, and then we build an index based on that. So for the remaining reads, we'll map against these junction libraries and then further figure out the BAM files, okay? All this code was written by Marcus Brace, and So this is the strategy that my lab is using. We know that we're missing something, but given the limitations of the current approaches, and I don't have a better way to do it. And another thing is for the novel exon discovery code. So based on the sequence rates to identify the novel exons. So we also have the code to do that, but it's not included in the general pipeline that we have. Okay, let's take another 5 minutes break.